Well, thanks everyone for sticking around for the session here today. We have one more presentation before the break at 3. Our next presenter is Alexey Markin of the National Animal Disease Center, USDA. And Alexey is going to present on making objective and informed influenza strain selections. So welcome, Alexey.
Thank you. Thank you. Can you hear me okay? Everything's good? Awesome. Thank you for the introduction, and thank you for the opportunity to present today. This is going to be another influenza talk, and indeed, I'm going to talk about a couple of computational tools that we developed at the Flu Crew at the National Animal Disease Center. And the first one is where we try to automate making objective selections for influenza A viruses and also beyond influenza viruses. Okay, I need to figure this out. So this is just to show you diversity of influenza A viruses. I'm sure many of you are very aware that influenza A viruses are very, very diverse. And so this is just the hemagglutinin gene. And we have two subtypes, as you know, that infect pigs, H1s and H3s. And so the different colors here on this phylogenies represent antigenically distinct clades. So this diversity groups that we recognize as separate viruses. And so you can see that many colors here that represent those clades. And so in general, at USDA, we typically see 14, 15 distinct clades every year. So there's a lot of diversity out there. And in addition to this, we have a lot of data coming out. So USDA influenza in swine surveillance program processes about 1,000 strains every year. And so there's a lot of sequence data that comes out of this. And so we need to be able to make objective selections. And so that's the question: how do we select representative strains from all this diversity and all this data? And that's something we wanted to answer. And there are two applications here. We want to do phenotyping of viruses with representation. And second, probably relevant to many of you here, is what do we put in the vaccine? We want to put representative strains in the vaccine. And so to me, this is almost like you're in a candy store, and you want to select everything, you want to characterize everything and protect against everything, but you only have $5. So you need to be really, really smart about your choices. And so the tool that we developed is called Parnas. And it helps you automate this process of selecting representative viruses. The input to the tool is always a phylogeny, like shown on the slide. Then we can ask Parnas, give us 3 most representative strains. And what it's going to do is something like this, where it simultaneously partitions your data, your diversity, into meaningful diversity groups, and then pulls out the representatives from within those groups. And so the representatives here are shown with the stars, and then what they represent is shown with the respective color. And so more generally, there are 3 slightly different sampling strategies that we implemented in Parnas. The first one is what you just seen, where we want to select a fixed number of most representative viruses. Second is where you don't know how many viruses you actually need or how many strains you need, but you have some other parameter in mind, for example, a diversity threshold or sequence divergence threshold. And then the third approach is where you combine the two. So I want to talk a bit more about the first approach, where we want to select a fixed number of most representative viruses. And so to illustrate how the algorithm works and potentially convince you that it's a good algorithm, so this is just a toy example where we have different strains here represented by different dots, and the distances between those strains represent the evolutionary distances. And then here we have 3 small diversity groups, and if we were to select 3 most representative viruses, then probably what you want is something like this, where you select strains from close to the middle of those clusters, where you're as close to everything else as possible. And that's the idea behind the algorithm. And so this is another example on an actual swine influenza clade. This is 2010.1, if that tells you anything. Again, here you see with the arrows the selections that the algorithm made, and then the algorithm also tells you what parts of the tree those selections actually represent. And this is just to mention, I'm not going to go into this, but this is just to mention that this is not just some heuristic that we put together. There is a very clear objective function behind the algorithm. So, and I'm going to spell it out, but essentially, given the phylogeny, you want to select k representative strains so that the average distance from all the other diversity out there to the closest representative is as small as possible. And that's pretty much the definition of representation in terms of the distance. And that's what the algorithm does. It always gives you the optimal selections under this objective function. Now to switch gears a bit, the second sampling strategy is, as I mentioned, when you don't know how many strains you need, but you have some other threshold in mind. The way the algorithm works here is that, so again, you have some diversity shown with the different black dots here, and then you have a radius that you're interested in, and this radius could be, for example, 5% sequence divergence or something else. And so you want to select a minimum number of strains so that all the diversity out there is covered within your radius. Like shown here. So those the 3 red dots here, they cover everything else within the threshold that you provided. And this is just to show how this works on the very small conservative threshold. Here we started with 12,000 human seasonal H1N1 HA genes. And then we downsample under a very small conservative 0.5% sequence divergence threshold. And then we go from 12,000 sequences to only 443 sequences, and we don't lose much information. We actually don't lose any information beyond that 0.5% threshold. And of course, 443 is not something you're going to stick in a vaccine, but you can play around with that threshold and get to the number that's actually useful. Okay, and so the final one, I don't have a good illustration for that, but as I mentioned, we can select a fixed number. So there are now 2 parameters, a fixed number of strains and also a diversity threshold. And so we want to select a fixed number of strains so that as much diversity as possible is covered under this threshold. And for that, I have a use case where we applied this to kind of a vaccine use case. The question was, how many strains do we need, given the surveillance system that we have, how many strains do we need to achieve high vaccine breadth, high vaccine coverage in terms of the number of strains that it can protect from? And the assumption that went into this, and this assumption comes from empirical evidence, that less than 5% divergence in the HA1 region of the hemagglutinin is usually a correlate of cross-reactivity. So often, if you have 2 strains and there's less than 5% divergence between them in the HA1, then probably vaccinating against one will protect against the other. And then we pick a year, let's say 2016. We look at the data in that year, the surveillance data that we had, and then we ask a question of how much diversity can we cover using 4 strains, 6 strains, and so on. And so this is a slide without results. Just to orient you, we looked at H1s and H3s separately. And for H1s, we look at how much diversity can we cover with 2, 4, or 6 representatives. And for H3s, where we have less diversity, we look at 1, 2, and 3 representatives. And then so we do selections in 2016, and then we look how long do those selections stay relevant over time. Then we also do selections in 2017, and again look at the same, and so on. And so when we do selections in 2016, for example, for H1s, you can see that 4 to 6 representatives across years give you, across the selection years, give you pretty high coverage of 80 to 90%. And then next year, typically that coverage remains relevant. And then maybe it drops off, starts dropping off after 2 years or so. And then for H3s, we have 2 to 3 representatives sufficient to cover 80 to 90% of diversity. And then that representation stays relevant for longer. So basically suggesting that perhaps 4 to 6 antigens for H1s might be sufficient. And then you would need to swap out those antigens every 2 years or so. And so this was an example where we did this assumption with less than 5% sequence divergence in the HA1, but we also wanted to do a next step and look at actual hemagglutinin inhibition data and maybe account for that. And so this is a study for human H3N2s, but I'm going to mention some swine data as well. So here we had the reason why we use human H3N2s is because there's really nice antigenic advanced predictions available in NextTrain for that. And so the tree that you can see here is 2,000 sequences, HA sequences from the 2022-23 flu season. And there are 2 vaccine strains that have been previously used, the Cambodia 2020 up there and Darwin 2021 down below. So many of you are probably vaccinated with some of those strains. And then when we use the 2 antigenic units, and I should mention that the branch lengths here, they no longer represent evolutionary distance. We rescaled this tree so that the branches represent antigenic advance. And that's why you have many of those gray lines where a lot of strains are on the same branch length. And so it turns out that those two strains, the vaccine strains, within 2 antigenic units do not cover everything, all the diversity out there. But we can use Parnas and ask, can we add a single new strain? And it tells you yes, they're shown with the red cross here. And then you achieve 100% coverage. So that's kind of thing that you can do. Unfortunately, we don't have really good antigenic advanced predictions for swine influenza yet. But what you can do is use directly hemagglutinin inhibition data and infer trees from hemagglutinin inhibition data or antigenic cartography. And so if you're comfortable with command line interface, I'd encourage you to play around with Parnas. And there is an associated publication in systematic biology. But now I want to switch gears and talk about reassortment some more, thanks to the previous speaker who set up the scene really nicely. So there's another tool that we developed for inference of reassortment from the surveillance data. And so the previous Chong did a really nice introduction into reassortment. So this is a silly introduction into reassortment, but it's essentially what it is, where, just a reminder, where we have 2 different strains co-infect the same host and the same cell potentially within the host. What you might get as a result is a kind of a hybrid virus that is reassorted. And so importantly, as you all know by now, a reassortment often seeds new clades and can change evolutionary dynamics of the virus. And so this example here is from Zeller et al. 2021 study where they looked at the HA-NA reassortment in swine influenza. And so here I think they have identified about 8 major reassortment events that seeded new major clades of neuraminidase, of virus. And so this is just to show you that reassortment is important. And so our question was, can we build a tool that can tell you when and where does the reassortment happen using the surveillance data? And importantly, we wanted to do this pretty much in real time. And so there are previous approaches that do this, and it turns out this is a very non-trivial question to do this. And there's even some of our work at the bottom, and there are some strengths and weaknesses of all of those approaches. But the main limitation for all of them is the amount of data that they can handle. And because of that, what people usually end up doing is just looking at a single gene segment, for example, the hemagglutinin, and track the evolution of the hemagglutinin. So we wanted to do something slightly different, where we let you look at the evolution of the hemagglutinin, but still map the reassortment events onto that evolutionary tree. And so this is just a small snippet of that. This swine H1s, just a small branch. And here you can see, so here you can see reassortment events mapped on top of this phylogeny. So and basically you read this as the hemagglutinin evolves from left to right, and then at some point over here, a new NS gene gets swapped in, and then we'll also tell you that this new NS gene was at least 38 nucleotides different from the parental, from the ancestral NS. And so this is over here, and then for this branch, you can see that there was a much more major reassortment event happening with pretty much the entire internal cassette being swapped in, and also the neuraminidase as well. And just looking at those numbers over there, you can see that these are massive changes. And so this is entirely new genotype. Okay, and so just to mention how this algorithm works, again, the toy example, so imagine that we have two strains, A and B. And when we look at the HA history, the evolutionary history of those two strains, we can infer that the common ancestor was about 1 year ago. So between A and B, there is 1 + 1, 2 years of evolution. And then we look at another gene segment, for example, the neuraminidase, and we see that between A and B NA genes, there is 50 substitutions. And then we can ask a question, is it likely that 50 substitutions have happened over 2 years of evolution? And we can apply a rigorous hypothesis test there because we know substitution rates for different gene segments and so on. And we can apply the hypothesis test, and if it's rejected, then we know that there was a reassortment between the HA and NA in this case. And that's essentially how the algorithm works. So this is, I'm gonna be quick here. This is to show that we did some simulation studies and saw that the algorithm is actually quite accurate. We simulated evolution with realistic parameters estimated from swine influenza. And we did see that whenever TreeSort reported a reassortment event, it was always correct, at least in the simulation study. And then in terms of how much of the reassortment the tool was able to recover, we saw about 80%. So the tool is biased towards reporting the truth. And we wanted that, so whenever it reports an event, we want that to be the truth. And so this is now when we applied this tool to the swine H1s. We had about 1,700 whole genome sequences, and this is, I think, pretty much all the whole genome sequence data that we had for the last 10 years in the US. And we detected 772 reassortment events, and those are mapped here with red circles. You can see where the reassortment happening, and the answer is everywhere, and it's really ubiquitous. And as a reminder, not all of those reassortment events have persevered, so to say, but a subset of those did create a new clade. And then this is just a comparison to what we get with human influenzas. Here we had 10,000 whole genome sequences of H1N1s, also in the US. And here we saw 253 reassortment events. And when we go back, we had less data, but many more reassortment events. So as you would expect, reassortment in pigs happens much more frequently than in humans. And another thing that we're going to do with this tool is look at preferential gene pairings and look at the biology of the virus. So I need to mention that you don't have to map reassortment onto the HA phylogeny. You can also look at other gene segments. For example, here we use PB2 phylogeny as a backbone, and then we map reassortment events onto the PB2, and then we can look at how often does PB2 reassort with other gene segments. And what we see is that, and perhaps expected, that PB2 reassorts least with PB1 and PA. And similarly, when we use PA as a backbone, we see that PA reassorts least with PB2 and PB1. And so that's perhaps expected, but the polymerase complex likes to stick together because probably when there is a reassortment happening, it breaks something in the polymerase. And so that's why you have preferential grouping of those genes. But they do like to reassort with the HA and the NA. And when we flip to the HA as a backbone, we do see that HA reassorts least with an NA, and NA reassorts least with an HA. So again, there is a preferential pairing there because we know that those two proteins need to work together. And so messing with those pairings can be not beneficial to the virus. This is the availability of the tool. And just to wrap up and come back to the Parnas tool, so we developed it for you to select most representative strains from your data. And then we showed that perhaps 4 H1s and 2 H3s can be sufficient to achieve high vaccine breadth, broad vaccine protection for about 2 years. And this study can be, of course, adapted to a specific farm or specific workflow, to specific flow and the data from that farm flow. And then reassortment is important because it can confound vaccination efforts, and it affects diversity and seeds new evolutionary dynamics. And then we developed a tool that helps you identify when and where does the reassortment happen, and that can serve as an early warning for a new genotype arising and potentially causing some damage. So finally, to wrap up, some acknowledgements. I'd like to thank everyone at the Flu crew. This is us. The group is led by Amy Baker over there and Tevis Anderson over here. And we also have collaborators here from the Iowa State University Computer Science Department, a group led by Oliver Ollenstein. And I'd like to thank all the veterinarians and producers who contribute to the USDA Influenza in Swine program. Without them, it would be impossible to do any of this work. And thank you. And I'd love to answer any questions.
Thanks, Alexey. We have time for questions, so we can look out here. Anybody, any questions for Alexey? One here.
Thank you for your nice talk. I'm Annie DeGroot from EpiVax, and I'm just wondering if you're talking about selecting the best representative strain, presumably you're thinking about vaccine development. When you select these strains, are you thinking about the antibody epitopes or the T-cell epitopes in the selection? So you really don't know whether there's an immune response that would be beneficial from the strains that you're selected.
No, yeah, that's a good point. That's not the only use case here, because another use case is phenotype and the viruses. But yes, a major use case would be doing vaccine selections. No, at this study we're just looking at the phylogeny of the hemagglutinin gene. You can account for other phenotypes. I'm not sure how to integrate T-cell response, but if you have some phenotypic data that you know of, you can integrate into the tool and inform the selections by increasing the weight of the strains that you know are beneficial to you and things like that. But generally, if you have, as I mentioned, if you have hemagglutinin inhibition data that could be useful for doing vaccine selections, then you can build a tree from that data, and then it would be informed by antigenic advance.
Right. Just to point out that Andres Gutierrez has a poster today that talks about the T-cell epitope content as a means of selecting strains, like similar to what you're doing, but slightly different.
Thank you.
Question over here in the corner here.
Thank you. Very nice presentation. This prediction is done in silico. You go to the pig, to the animal model to see if your predictions are correct?
Do you mean for the reassortment?
Yes.
For the reassortment.
And for the prediction for the vaccine that you check?
Right. No, so we haven't. Thank you. We haven't done any studies to follow up. I don't think there is any vaccine trial where vaccines were selected using the tool. We're open for that, but it hasn't been done yet. For reassortment, we have empirical confirmation that the reassortment that we detect is the truth just from the surveillance data, because we can look and we see a different genotype happening, and so there we do have clear confirmation for Parnas. It's a little bit more difficult to say that this will be for sure a great vaccine, but maybe it will be, maybe it won't.
One more question, Kim.
All right, thank you. Kim Vanderwall, University of Minnesota. Very interesting talk. I am intrigued by the tool tree sort, and I think that there's certainly major advantages in terms of not actually having to have the parental strains in order to actually be able to detect the recombination event. So I think that that's a nice advancement for incomplete whole genome surveillance data sets. But my question, I guess, is how is this really something that's been developed only for influenza, or is it fairly easy to apply to other viruses?
Yeah, thank you. That's a good question, I'd imagine you're probably thinking of recombination in like PERS or something like that.
I'm thinking of PERS, but I'm also thinking about other segmented viruses.
Yeah, for segmented viruses, for sure, that would work. For something like PERS, I'd imagine because you can have within gene recombination, one approach that you can probably use in PERS is just somehow chunking up the genome into 100 nucleotide sequences or something like that, and applying this approach, assuming that it's going to work well, or maybe first scanning for where does recombination happen in PERS and selecting the chunks of the genome that are non-recombining, and then using those as segments for tree sort or something like that. So I think it's doable, but it is messier because, segmented virus is much cleaner that way, and it's easier to model.
All right, I have a lot of other really technical questions that I'm not going to ask right now, but I'd love to talk to you at the coffee break.
Thank you.
Well, one last question here, Matt.
So great talk. Thank you for bringing these new tools so we can use them. I'm just curious, these are local programs, command line interface type. Is there any thought of moving these products to a publicly available GUI type system, maybe like a BVBRC workspace that people could potentially load their own data sets in and not necessarily have to have the computing background to make these types of calls?
Yeah, thanks. Yeah, that's an important question. And for TreeSort, we are collaborating with BVBRC right now, and hopefully at some point it's going to appear there. But probably there is a way for us. It's an investment of effort and time because it's hard to build GUI. If there is interest from the community, we can do that. Yes.
I lied. We have time for maybe one more question, so we'll keep this as our last one here, and then we'll wrap up.
So, just quick question. I'll follow up with the other gentleman question over there. I believe you are doing the prediction based on the HA sequence, right? And you may not have to do the protection, but can you comment on the association between the distance in the vaccine that you select and the HI titer, because that's what more simpler to do than the protection.
Right. Thanks. So the only people look, of course, at there are models that look how much hemagglutinin inhibition data correlates with evolutionary distances, and usually it does correlate. And as I mentioned in one of the slides, we typically see empirically that about 5% divergence in the HA1 region of the HA in the amino acid sequence usually tells you that less than 5% divergence tells you that there is some cross-reactivity remaining, and so we use that to inform our selections. But broadly, of course, a single amino acid change can be a massive change, and so normally, ideally, you would use more advanced models of an antigenic advance to do those selections, and we have that for human data, but we don't have that for pigs just yet. But we're working on that.
Great. Thanks, Alexey. Give Alexey a round of applause. That concludes our session here. There are refreshments. Let's give our three speakers, one more round of applause. Thank you. And thank you for attending the session, and enjoy the break.